Consider the following diagram of double-stranded DNA. The red strand, we will consider the top strand with an orientation of 5' on the left, 3' on the right where the arrowhead is. Because the blue strand is the complementary strand, we have it oriented in an anti-parallel fashion with the 5' end on the right and the 3' end on the left. Now we could zoom into some of this sequence and visualize the base pairs. If, for example, the top strand reads A, T, T, A, G on the top strand, then the corresponding sequence on the bottom strand would be T, A, T, C in an opposite orientation. Now let's imagine that we want to amplify the region demarked by these black lines using PCR. We'd need both a forward primer and a reverse primer. Let's think through the forward primer first. The forward primer would bind in a 5' to 3' orientation to the bottom strand. Therefore, it needs to have the sequence A, T, T, A, G in a 5' to 3' orientation to complement this bottom strand. This sequence is identical to the sequence of the top strand because both of these sequences are complementary. Now that we've defined a forward primer, we need to identify a reverse primer that will amplify the top strand from the other direction. So let's again put in some arbitrary sequences so that we have some base pairs to think about. From 5' to 3' in the top strand, let's make this A, T, G, C, and in the corresponding sequence on the bottom strand would therefore be T, A, C, G. Once again, the primer 2, reverse primer, would need to bind to the top strand to match the forward primer so that both strands are being amplified during this PCR. That means that the reverse primer needs to base pair with this top strand. Its sequence, therefore, would be G, C, A, T. Because it binds in an anti-parallel fashion, we're going to have the 5' end on this side, 3' end on that side. Therefore, when that primer gets written out, convention dictates that we write it from the 5' end first, G, C, A, T, ... depending on how long our primer is. During the denaturing step of PCR at 95 degrees Celsius, the complementary strands of the template DNA will separate from one another. Then the reaction is cooled to 53 degrees Celsius in our experiment to allow time for the primers to anneal. Note that the primers exist in the PCR solution this whole time. They simply are dissociated from their complementary sequences until the appropriate temperature is reached. Next, the temperature is shifted to 72 degrees Celsius, activating Taq polymerase so that each primer can be extended by the enzyme, copying the complementary sequence. The amount of extension that takes place is dictated by the amount of time that the PCR reaction is held at 72 degrees Celsius. We can imagine that this is what the DNA products look like at the end of round 1 of PCR. At this point, I encourage you to pause the video and draw out for yourselves what round 2 and round 3 of PCR might look like. if you need to check yourself, this is what round 2 would look like, the end of round 2.